I'm Alex Popham. I'm the 4-H agent here in Breckinridge County, part of the University of Kentucky's College of Ag, Food, and Environment's Cooperative Extension Service. And today we're continuing our exploration of science, engineering, and technology by extracting some DNA from some bananas. This particular lesson is from our 4-H Biotechnology curriculum, and I'm just going to walk you through the steps today. So to start off with, I just blended 2 whole bananas, peeled them and put them in the blender with 1 cup of water. Distilled water is best. And then I blended it for about 15 seconds. And then after I finished that, I set it to the side and I filled some vials up with alcohol about 3/4 of the way. And I've just had them sitting in ice so that they'll get nice and cold. The next thing you'll want to do while that is cooling is in a 5-ounce paper cup, you need to add 1 spoonful of shampoo. Now this particular shampoo is strawberry scented and I stole it from my husband, so thank you, Jacob. To that 1 spoonful of shampoo, I'm going to add 2 pinches of salt. You'll notice these are all things you can find in your own home. So this is a great science experiment to do with your family. And then I'm going to add 4 spoonfuls of distilled water. And then you'll just gently stir that. You'll tell that I'm not stirring very vigorously. I don't want to stir so hard that it gets super foamy. All I'm wanting to do is dissolve that salt and dissolve the shampoo. While I'm doing that, the purpose that we're adding the shampoo to our banana mixture in a little bit is to help break those cell membranes. We want to break the cells of our banana open, and the soap will help break down those fats and break into not only the cellular membrane, but that nuclear membrane that holds the DNA. And then we added a couple pinches of salt because that's going to help hold the DNA together and help it clump and keep those phosphates that are part of the sugar phosphate backbone from breaking down. Try to keep the DNA all together. Once you get to a point where you feel like the salt and the soap have dissolved, then you're going to add 2 good spoonfuls of your banana water mixture. Smells so good. I love banana. So 2 spoonfuls was all it took. And then you're going to stir this for about 5 to 10 minutes. Now I'm not going to keep you on here for 5 to 10 minutes watching me slowly stir this mixture, but during that 5 to 10 minutes, that's when those cell membranes are breaking down and the DNA is starting to kind of cluster together, and that salt's helping shield the phosphates that are part of the DNA molecule from getting damaged. So I would just continue to do this for 5 to 10 minutes. We're going to pretend that 5 to 10 minutes have passed. And the next step would be to filter. So now we've broken our banana cells up, but we only want to keep the DNA. I want to filter out all the other components of the cell that I don't want. So to filter, all you need is a simple coffee filter that most of us have around the home and a paper cup. About 5 ounces is fine. And then I'm going to use some paper clips to help me out because I don't want the filter touching the bottom. I'm going to leave space for the mixture with the DNA to drip through. So I'm just going to push it partway through, just a little bit. And then I'm going to fold it over the edges of the cup and paper clip the sides. So you can see I've created a filter, very cheap, inexpensive, easy to assemble. And the paper clips are my own addition. I just like not having to hold the filter paper in place, so the paper clips do that for me. After I had mixed that solution for 5 to 10 minutes, then you're going to add it to your filtration system. And it's going to drip slowly. It'll probably take 2 to 3 minutes to fill the bottom of the cup or cover the bottom of the cup with some filtrate that contains your DNA. Once the bottom of the cup is full of that DNA solution, then you're ready to collect it with a pipette. So I'm going to stick my pipette down in there while I go grab my vial of alcohol that's been cooling. So again, I filled it about 3/4 of the way. So I have my test tube, I'm sorry, not pipette, but my test tube filled 3/4 of the way. And then I'll just add a test tube or a pipette full of our DNA mixture directly into the alcohol. And the hopes is after 2 or 3 minutes, when we add that solution with the DNA is then going to precipitate to the bottom of our test tube here. And you can use any kind of clear container. You don't have to have a test tube at home to do this. For me, it just made it a little easier because once you let it precipitate for 2 to 3 minutes, you don't want to shake it, you don't want to mess with that test tube, just let it precipitate and that DNA separate out. When the DNA separates out, it's going to look white and streamy, and you can even use a toothpick to pull that DNA out and play with it. And that's the DNA that you're eating when you eat your banana. And I do have a test tube that I've already extracted the DNA from. And you can see all that lovely white banana DNA that clumped together because thank goodness we added the salt. And it's just DNA because that shampoo helped break apart the cell and the nuclear membrane, and we were able to filter out everything else except the DNA. So now it's filtered in that cold alcohol solution, and you can play with it and get to see that DNA for yourself. So try this at home. All these supplies are probably readily available in your kitchen, your bathroom, and have fun with it. Enjoy. Thank you.